I'll be introducing our second speaker. So our second speaker today is Dr. Roopali Chaudhary. She is the founder and CEO and lead researcher of the nonprofit Lotus STEMM, an organization geared towards STEM outreach and increase the visibility and representations of South Asian women in STEM fields. She is also a senior scientist of cell biology specialist with Allarta Life Science Inc. and a research associate with Dr. Juliet Daniel at McMaster University. Dr. Chaudhary changed fields and research specialization after each of her degrees. She started university with a desire to enter the medical field, but realized her love for research. She began with working on fruit flies, switching to studying inflammatory bowel disease using a mouse model, followed by immunology of food allergy. Currently, she works on long-term therapeutics of type 1 diabetes. In her free time, Dr. Chaudhary works at a custom cake bakery, focusing on science-themed cakes and enjoying clay sculpting, woodworking, and exploring building furniture. Please give Ms. Chaudhary a warm welcome.
Hi, everyone. Can people in the back hear me? Yes? Awesome. Thank you. I have just realized that when I created these slides, I completely covered the last letter of my name. Oops. Anyways, as I was introduced, my name is Roopali Chaudhary, and I thought I'd start off first giving a little bit of background of who I am and how I ended up where I am now. So I was born in India, in New Delhi, and we lived in multiple countries. I was brought up mostly in Middle East, so I lived in Kuwait and in Saudi Arabia, and I have gone to school in India as well. And throughout my childhood, I was so determined that I was going to be a doctor. When I grew up watching Bollywood movies, okay? And growing up, I used to see the women were usually nurses, and I was like, I want to be able to help people. But I don't like taking orders from anyone. So I should be the doctor instead so that I can boss people around. And that was my reasoning for why I wanted to go to medical school. And all throughout my high school, I was so determined. Med school is what I want to do. So I moved to Canada when I was in grade 8. And I finished my high school here in Canada. I did it in Waterloo. My parents still live in Waterloo. I go visit them very often. And then from Waterloo, I was like, now what? Well, to get into med school, you should get a science degree. That means go do a BSc at a prestigious university. So I went to University of Toronto, St. George campus, not UTM. And here, this is the only university that did not give me a scholarship. And I don't know why, but fine. I still went there. And I realized within that first year that I was way over my head. I did not know what university is supposed to be like. How am I supposed to study for this? And it's a little bit of a shock when you go to university. So anyone who's in like high school, if you think you know what's coming, you don't. And that's okay. That's what university is supposed to be. It's supposed to push you outside of your comfort zone. And that's what it did for me. So I realized in about second year, I got into the specialist for molecular genetics and molecular biology. Long name, I had no idea what that even means. I just heard specialist. Yes, this is going to get me into med school. So I'm going to do it. And up until then, I had no idea that there were scientists that are still alive. I thought they were all dead and gone, because I'd only ever learned of like Einstein and Newton, and they were all like long dead. So I didn't know that my professors were also scientists. And this is something that intrigued me. I was like, oh, I don't know, what does a scientist actually do? So in second year, when we started to have more hands-on labs, I learned research is like solving puzzles. And I like puzzles. So maybe this is my calling. Maybe I don't want to go to med school, but I still want to help people. So how do I do that? And I'll give a very quick story. So I was in my genetics lab, and I learned of this scientist called Barbara McClintock. I had not heard of women scientists before that. So this was just eye-opening for me. This is also like internet and smartphones was not a common occurrence. There were no smartphones. What am I saying? There was no smartphones. We had just gotten internet in our residence too. So Barbara McClintock popped up and she discovered these genes that are called jumping genes or transposons. And she studied corn. She looked at, there's your normal yellow corn that we all eat, right? Everyone's just like staring at me, so I'm trying to get some nods. Yellow corn, you guys recognize those. And then there's corn where you can have the brown color kernels and sometimes orange and red, and some are like really deep purple. But the reason that happens is because genes can jump from one site to another, and that can lead to a change in color in the kernels. And Barbara McClintock is the one that discovered this. She went on to win some very prestigious prizes, and this information changed the way we understood how cells can behave. And this was eye-opening for me. So I was like, I'm going to go and do research. And it was really hard to find a position. I was not a very good student in university. I will be upfront about that. And again, that's okay. I found a position where we were studying fruit flies, Drosophila. Who knew you can do a lot of research on fruit flies? Show of hands. I see a couple of hands. Yeah, those fruit flies that annoy you in the summer and you're trying to kill them in your house. I have the trick. If you want to know, come talk to me after. But I decided I have this opportunity. I get to learn how to work with fruit flies. And it was one of the most amazing experiences. Mind you, I started off cooking fly food for them. And if you don't know what fly food is, which you probably shouldn't, because you don't work in a fruit fly lab, but all it is cornmeal with some yeast and some sugar, and you boil it and mix it and pour it into these test tubes, and the fruit flies love it. So I learned how to cook that first. That's what my first few days in a lab was. Not the glamorous, I thought I was going to have lab coat and wear goggles and do all this fun stuff. It's not what it was. But I learned so much about what does it mean to do research. So I decided to do my master's. And I did that at University of Waterloo. Remember, up until this point, I was very much like med school, and then undergrad, I had this eye-opening, and I had to talk to my parents and let them know that I didn't want to apply to med school anymore. I wanted to go to do my master's. I am Indian, and for anyone who has ethnic parents who are very determined that you need to be successful, may believe that the only way to be successful in science is to go to med school. No, you can do other things. And I had to convince my parents that it was okay for me to go to graduate school. So my master's, I actually studied the embryos of these fruit flies and development of these embryos. And you're probably wondering why. What's the impact of that? What are you going to learn from that? Well, fruit fly embryos, they have this extra tissue that helps protect the embryo while it's developing, but it's programmed to die at a specific point. And that programmed death, if we can understand how cells are pre-programmed to do that, maybe we can actually extrapolate that and use it towards cancer cells. And that was cool to me. And I learned you can have a lot of signaling between these cell types. They give each other, same as how you communicate with your friends and your parents. That's how cells communicate. They talk to one another, and that makes the cells then can make decisions on where and what they want to do. So that was great. Okay, now what? So I wanted a different experience. And there was a speaker that came to University of Waterloo, gave a talk, and she talked about how she discovered this protein, and she named it Kaizo because she used to listen to a lot of calypso or Kaizo music. when she used to do the experiments. And so she decided to name it after the music. So she called it Kaiser. And I was like, she sounds like she's fun. I want to go work for her. And so I went on to do my PhD. Now my PhD, I switched fields. It wasn't on fruit flies. Now I was actually going to be working with small animals. And I had to create and characterize a genetic model and understand how does a phenotype develop. And the phenotype that I discovered was inflammatory bowel disease in these animals. There have been no really good genetic models for it that allows for spontaneous development. So our model became really good. We can understand how the immune system works, how your epithelial cells in the villi of your intestines, I'm saying very big words, I'm going to come back. But the idea is, how does your immune system and your intestinal cells work together? And what causes the inflammation? So in humans, what is leading to it? Are there genes behind it that can impact? And that was very exciting. But then my PhD came to an end and I needed to do something else. So I did what's called a postdoctoral fellowship. This is like it's getting you ready to become a professor. So you're going in, you're doing a lot of research. And I switched fields. This time I decided, let's go learn about the immune system. And I'd never taken a course about the immune system, but I decided we're going to do this. And I researched the immune system's response to food allergy very early. So in humans, you learn that you're allergic when you have an allergic reaction. You don't know before that if you are or if you aren't. And we don't know what causes humans to become allergic. So what I was studying was that before you know that you're allergic, what is happening? That's where my research was. I did that for like 3 years and I needed a break. So my break was, I'm going to teach for a bit. And so I taught cell biology course, but I also started Lotus STEMM. And this was a big step for me. Up until now, I'd not done anything that was business related. And Lotus STEMM as a nonprofit, I had to learn how to be an entrepreneur, how to do accounting. I didn't do accounting. How to do social media. Do y'all know how hard social media is? God. Anyways, but I had to learn all of that. So it was a great opportunity. I did many other things. I've written as a content editor. I taught again. But where I am now is I look at your pancreatic cells that secrete insulin and how can we protect them when we do long-term therapeutics for type 1 diabetics. This is where I am right now. But what I really want to summarize about this is between kindergarten and grade 12, I went to 10 different schools. I've lived in 4 different countries. I've gone to 3 different universities. I know a lot of techniques. I've done many disciplines. And I run a nonprofit, a bakery, and I love woodworking. And at the end of the day, what I want you to realize is what I thought I was going to do is not what I'm doing right now, but I'm still doing it in a way where I'm helping people. And that was my initial goal. So your path is going to change, and that's okay. It's okay for it to change. You cannot have everything pre-planned. You just have to figure it out. So I was also asked to talk about cell biology and why it's important. I'm going to throw this back at you guys. Name as many cells as you know. Just shout it out. Nerve cells, red cells, skin cells, blood cells, stem cells. White blood cells, red blood cells. I'm hearing a lot of blood cells. Bone cells, okay? Yep. Eukaryotic cells. Immune cells. Okay. So, if you didn't notice, all of you guys, when you started to shout out cells, were shouting out cells that you know in your body. But there are so many types of cells. You've got what classifies as prokaryotic versus eukaryotic. You probably have learned this in school. We've got bacteria. There's different types of bacteria. And bacteria are different types of cells. We've got fungi. Fungi are made up of cells. You've got animal cells. You've got plant cells. Anything that's living has got cells in it. Amoeba is a cell. And because the living world is made up of cells, when you want to solve a problem for the living world in some way or form, you need to understand cell biology. So I'll give a few very quick examples. We've all heard that there is a global climate change that's ongoing. We've heard of microplastics that is in water. We've heard that there's a lot of CO2 emissions that have increased. Well, cell biology, where it's going right now, is creating lab, quote unquote, meat, so that we don't have to cultivate large land for animals. There's also ways on improving agricultural methodology so that plant cells can actually be more sustained in the global climate that is continually changing. There's also ways that we're looking at how are microplastics in the water impacting aquatic animals and humans. So what is the interaction with cells? What's happening there? Cell biology, very important. So if your question is global climate change, what is the impact? In some way or form, cell biology will fall in there. There's also chronic diseases, a lot of chronic diseases in humans, in animals, in plants. These can be solved by understanding cell biology. How are cells interacting with one another? And based off that, what can we do to improve it? There's cell biology research going on the International Space Station. So we're talking about actually going outside of the Earth and exploring. We need to A, survive that, B, be able to grow food, C, make sure that we have bacteria that can survive with us because we need the bacteria in our body. And we also need insects because they're pollinators. So how is everything going to survive? And understanding what happens in space to cells is really important before we can actually start doing long-term space travel. But let's think more deep. There is also deep sea creatures. And these deep sea creatures have very different types of proteins that we may not necessarily see in our cells. And so can we use those proteins for stability of perhaps vaccines that need to be delivered to all parts of the world? So it depends on the question, and there is ways that cell biology would get involved as long as you're looking at the living world. The only thing I'm going to say at the end here is I hope my talk really did show you that don't worry so much about what you want to be. Think about what do you want to solve and how would you solve it? That is a model that you can actually have throughout life. With that, I say thank you. We do have a booth outside. We have an aerospace engineer that has joined us with a 3D printed landing gear demo that she'll be doing. I'm happy to take any questions. Yes. Could I just pause you on that? I am a cell biologist. If you're going to ask me about engineering, I'd say, come to our booth and talk to the aerospace engineer. Got it? Okay. Go ahead. Oh, how do you get rid of fruit flies? So there's a few things fruit flies love. One is fruit that is overripe. So you need something that's going to ferment, and they like sugar. So what you do is, in a bottle, put in water, put in a little bit of yeast. If you don't have yeast, do apple cider vinegar, like a tablespoon, and do a drop of soap. That just breaks the surface tension on the liquid, and put a funnel in that bottle. Fruit flies will love the smell of the apple cider vinegar or the yeast. They will fly in, and because it's a funnel, they won't be able to find their way back out, and you'll be able to catch a whole bunch of fruit flies and clear out your kitchen. Yeah. Oh, how do you get rid of zoonotic diseases? Can you give me an example of one? COVID-19. Okay, let's talk about COVID-19. I'm going to keep this very brief because people do not like hearing about COVID as much. Now, COVID-19, fascinating virus. As a cell biologist, the theory behind it is really interesting. Having lived through a pandemic, the reality is very different too. How can you get rid of it? It is very difficult to completely get rid of it because we live on Earth, evolution is going to happen. Viruses, bacteria are going to continue to evolve, and we continue to evolve throughout time as well. Humans have been continuing to evolve as well. Are we going to get rid of them altogether? No. What we can do is understand what the impact is, and using our cell biology knowledge that we've had for years, decades, we can actually apply that. So the mRNA vaccine, for example, that was given out for COVID, the research has been backed up for vaccinations for HIV, actually. So it's not like brand new technology. It was just technology that people didn't know about. However, we back everything up with research. And using decades of knowledge that we have and the scientific methodology, that's how we approach it. We won't get rid of it. Yes. Oh, what a fascinating question. Okay, so the question was, why are scientists using induced pluripotent stem cells instead of embryonic stem cells? First and foremost, how many people know what induced pluripotent stem cells are? A couple? How many know what embryonic stem cells are? A few? Okay. So embryonic stem cells, can someone tell me where embryonic stem cells are found? Yep. Not necessarily in the fetus, it's actually in the umbilical cord. So the umbilical cord, when a baby is born, is more or less tossed out. And we can actually isolate the embryonic stem cells from it. But people, people don't like the idea of it. However, they're also difficult to work with. Induced pluripotent stem cells are, you can take cells and revert them back to being stem cells, and you can induce what type of cell you wanted to differentiate. So, for example, stem cells can become any kind of cell in your body. Most of your tissues that have continual growth will have some level of stem cells that will continue to differentiate and become that cell type. With induced pluripotent, it doesn't have to be in that specific tissue. So you can actually say, I'm going to take stem cells, induce this to become kidney cells. Or I can induce it to become an immune cell type, or induce it to become a insulin-producing islet cell. And when you do that, you actually do broaden up where you can use the induced pluripotency. You can actually take stem cells from the person and you can revert them back and then induce it to what you need it to be. So in terms of therapeutic, it's a little bit more specific to the person as well, as opposed to embryonic stem cells. It's a very general answer. I can't go into too much of the detail because it might be need to be at a higher level because this is like master's level questions. Yes.
Would you say when you study like fruit flies, if we eliminated fruit flies from our ecosystem, would that have any negative impact?
Oh, that is another fascinating question. So the question is, would there be a negative impact if we eliminated fruit flies? Now, fruit flies are part of the ecosystem. There are other insects that survive on fruit flies. So would it have a negative impact? Probably a little bit, not enough for us to notice. They are a little bit of a nuisance, but they're not necessarily spreading diseases. So it's not like we really need to put in the energy to get rid of fruit flies. Now, mosquitoes is another question. And there is research going on can we completely eliminate mosquitoes or make mosquitoes so that they can't actually bite you or cannot spread disease such as malaria. So it's always a pro and con. Do you need the effort and the energy to eliminate fruit flies versus something else that could actually lead to diseases? Yes.
It took somebody from 10,000 years ago and brought them into today. How dangerous is society these flies?
Very, actually. So from 10,000 years ago to now, if you think about how much has changed in terms of not just healthcare, but in terms of how our bodies have adapted to the food we eat, to the interactions that we have with our environment, to the different type of pollutants that are in the air. So based off all of that, taking someone from 10,000 years ago, they'll have better medical care now, but their cells may not actually be able to completely adapt. It'll take some time. It won't be like they'll just automatically get a disease and die, but it would be a shock to the body itself because they won't be used to the pollutants that we have in the environment compared to what it was 10,000 years ago. They would not be used to the food we eat now because, believe it or not, plants also continue to evolve and evolution has happened naturally and has been happening ever since humans have been doing agriculture. So based off that as well, there's probably difference in the nutrients that they would be getting. And we have different diseases now than what were around in 10,000 years ago. So that probably the body would have quite a shock. Yes. Okay, so the question is, if fruit flies get trapped in the bottle, how do you get rid of fruit flies that are in the soil of the plants? Now, if you actually do create the fruit fly trap with the funnel, fruit flies are not smart enough to find that hole again, and so they won't come back out. But the thing with fruit flies is they lay a lot of embryos. And the females can store sperm in them for about 10 days and continue to lay fertilized eggs everywhere. So it all depends on when you actually caught them. Now in soil, you may actually just need to put one of those sticky traps, you put that in, and then there's something called diatomic earth that you can put that will actually help dehydrate those embryos and the larvae so that you don't get more adult fruit flies flying around. But the best thing is just have a little bottle with a funnel with the water, the apple cider vinegar, and a little bit of soap. That will help clear out the air a lot. Let's go here. That is an excellent question. So the question is, at what point in my life did I decide to study cells? It was not until I had finished second year of undergraduate. When I went into university, I was still very determined that I'm going to go into med school, and I planned it all out and what courses I needed to take and which med schools I was going to apply to. And I finished second year. I wrote my MCAT, and I realized at that point that the topics that was interesting me was how cells communicate with other cells. So the cell signaling that happens. And at that point is when I realized that maybe I don't want to be in the hospital as bedside physician. I want to help, but I want to help in a different way. The puzzles that comes with it is what really intrigued me, and I get very excited when talking about cells, and everyone in my life knows this. And so it's where I found that joy. It was not easy though. I didn't always love it. Yes. Yeah, that is an excellent question. So the question is, how are scientists working on eliminating mosquitoes, and what's going to prevent mosquitoes from evolving to become immune to whatever scientists are doing? What scientists are actually doing is they're making the male mosquitoes so that when they mate with a female, it actually prevents her from being able to lay embryos. So the male mosquito becomes infertile pretty much, and the female cannot lay embryos. So that prevents that next generation of mosquitoes to come. They've actually released some of these mosquitoes, I think off an island somewhere in the Atlantic Ocean. I don't know where exactly, but there is pilot experiments happening on if we do release it into the wild, are they going to spread and prevent mosquitoes everywhere, or do they stay? And right now what they found is they stay around that island. They do not have the capacity to like go worldwide. So they would have to release in like multiple places. And what's going to prevent mosquitoes from evolving to be immune to it? Nothing. Nothing can prevent evolution. Evolution will continue to happen, and we may get a different type of mosquito. It just wouldn't be the species that we have. And then the question would be, does it actually still spread diseases the way the current mosquito species do? Yes, in the back. So let me come up. Can you induce cells to replace cancer cells? So another excellent question. I love the questions here. Guys, it's so good. Ooh, pinch all your cheeks. Okay, so the question is, can you induce cells to replace cancer cells? There is so much research going on how do we target specifically cancer cells and not impact your healthy cells. So when you go through chemotherapy, it is killing any cell that will quickly divide, and that's cancer cells, they quickly divide. But that's why you start to get stomach issues, because those cells are quickly dividing in your intestine, your hair, your skin. These are like fast dividing cells. So there's research going on like how can we target specifically the type of cancer that a person may have? How do we kill that so that your normal cells can grow and take up that space? And there are ways that people are doing, and this is called personalized medicine. It's taking a biopsy out of the person, understanding the characteristics of that type of cancer cell, and then how do we target it? So we actually create immune cells that are targeted specific to that cell type, increase those immune cells and put them back into the person so that you can start to kill them. Now, we're not at the point where the personalized medicine is something that's available to everyone, or that it's like 100% efficient, but we're getting there. We're getting very close. The immune system and cancer cells and your regular body cells are very complicated. So trying to make everything work at the same time is a little bit difficult, but we are working towards it. And I think in the next 20 years, we'll be talking about personalized medicine a lot more. With it. Before I take more questions, I need to check in with the team. How am I doing on time? We're going to wrap up. I will be at the booth. If you guys have more questions, please come and talk to me. And our aerospace engineer, Ms. Juliet Pinto, is going to be there with the landing gear. I'll talk to you all soon.
Thank you, Ms. Chaudhary, for that presentation. We have a gift. Sorry, Dr. Chaudhary from the presentation.
Thank you.
Here's a presentation, or here's your gift. Thank you.
Thank you very much.